Hello guys, I'm Rishabh, and today we'll be starting with the discussion week 5 for the RNA biology lectures. And in the last class, we discussed about the properties of the genetic material. So, what are the properties? It must be capable of replication, that is, it must create its own replica. Okay, this was the major criteria for a molecule to be a genetic material. It should be stable both structurally and chemically, because if it is not have a stable structure, then it would be prone to degradation. And if it is not stable chemically, then it means that it is very reactive, and it must have a scope for slow changes, that is mutations, to evolve, so that with time it must evolve. And it must be expressed in the form of Mendelian characters. That means it must show the phenotypic response, and it must pass stably through the generations. Okay, so for example, if this is the genetic material, then it must pass through from parents to offsprings. Okay, so these were the some properties of the genetic material that we discussed about. And the major uh, question is that whether RNA can be a proper genetic material or not. So in today's reality, RNA is not a perfect genetic material. Okay, but in some viruses, it is being seen that it is the genetic material, because it was primitively known to be the genetic material according to RNA world hypothesis. So again, what is RNA world hypothesis? That RNA has a catalytic role, and it has self-replicating capabilities. Okay, so based on this hypothesis, RNA was earlier the considered to be a genetic material in the early organisms, and further from RNA, DNA was originated. So, what are the properties of RNA that does not make it as a good molecule as a genetic material? So, it is less stable because it is highly reactive. So, as we discussed, it has a 2' OH group. So this is the first carbon, this is the second carbon which has a OH, this is the third carbon with the OH 3 prime, 2 prime, 1 prime, this is the fourth carbon, and this is the fifth carbon through which phosphate group is attached. Okay, so here the base is attached, and this is the 2 prime OH that makes the RNA reactive. Okay, as you have all seen in various ribozyme molecules also, this 2' OH goes and binds to different molecules, forming various phosphodiester bonds. So it is a nucleophile because of the lone pair present on the OH group. Okay, then it is more prone to mutation, that is, it evolves very faster. As you can see in various viruses, for example, we can take the recent example of coronavirus. As you have seen that it evolves very faster. So one type of vaccination could not work on this because of the evolution factor of these viruses. So because of their highly reactive nature, they are more prone to mutation, and that's why they evolve very faster, and they express in the form of Mendelian characters faster than DNA, because as we know, the central dogma states that DNA first converts to RNA, then to proteins. Okay, So for DNA to form the proteins, RNA is the intermediate. But if the RNA is the genetic material, then there is no intermediate. Directly the proteins are formed. So here the expression is faster as compared to DNA. And as I have discussed, it is genetic material of some plant and animal viruses. So in plants we discussed about tobacco mosaic virus, and in animal viruses we discussed about the HIV, or the human immunodeficiency virus. And RNA, being the genetic material, replicates either through uh, RNA dependent polymerization, or through DNA dependent RNA polymerization, that is through reverse transcription. Okay, so these were the points that we discussed in the last class, and today we'll be starting up with the transcription. So, as RNA is not a good genetic material, so what is RNA used for in the molecular biology system? So, as RNA has certain properties of being reactive, and like it is directly encoded by the protein, so it is considered as a molecule that helps in transmission of the genetic information. So all these properties stated above makes it a good molecule for transmission of genetic information, as compared to storage of the genetic information. Okay, so that is why RNA is expressed in the form of proteins, and this is we'll be discussing in the central dogma. So we'll be starting up with the transmission of genetic material information through the central dogma. So central dogma was basically given by Francis Crick in 1957. And what is the dogma means? Dogma means a statement that is not proved by a proof. So this is a statement that is being not like hypothesized with the proof, or with some of the information, background information. Okay, so that is why it is called as a dogma. So this basically tells how the phenotypic characters are expressed. Okay, so what it states is that DNA, it can produce DNA through replication, and then this DNA transcribes the genetic information in the form of the mRNA, that is the messenger RNA. And on messenger RNA we have triplet base pairs, that is called codons, that we'll discuss when we'll see the translation process. So in the codons, that one codon code for one amino acid. So this mRNA is now converted to this information is converted to the proteins. Okay, so this process is known as translation. So first DNA transcribes the information through mRNA, that is called transcription, and mRNA is then formed in the form of proteins, that is called as translation. So this protein molecule now folds and functions, or express the phenotypic information. Okay, so this is basically what is called as the central dogma. And there is one more concept that was given by Temin and Baltimore. So what this states that in some viruses, so they saw majorly tobacco mosaic virus. So what they saw was कि in some viruses this mRNA is reverse transcribed to DNA. Okay, so this is called as when this mRNA is converted back to DNA, this process is called as reverse transcription. And now this DNA follows the same process. So it first converts to mRNA through transcription, then mRNA forms proteins, or this particular mRNA is like packaged into nucleic acid. So this process, that is Temin and Baltimore, that was observed is particularly seen in HIV that we have discussed in the last lecture. Okay, and this particular process is called as reverse central dogma or Teminism. Okay, so this was the concept of the central dogma that shows that how the information is transformed from transmitted from DNA to RNA and then from RNA to proteins. So now moving on, so we'll discuss a bit more detail about the transcription and translation. So first, what happens? This is the DNA that we are talking about, and here we have two strands. One is the coding strand and one is the non-coding strand. So we discuss what are these. So first, what happens? The uh, DNA dependent RNA polymerase, because it DNA is the template, and what is being formed? RNA is being formed from being DNA as a template. So now when the polymerase, so anything that we are forming is formed by the help of polymerase. So now what we are doing is, here DNA being the template is being acted upon by the polymerase to form the RNA. So hence the name DNA dependent RNA polymerase. So now this particular enzyme is working on DNA to form RNA, and here the RNA chain is being formed. Okay, and this generally what happens, one strand of DNA. So here only the one strand, that is non-coding strand, is being used to form the RNA. And the concept of coding and non-coding we'll see when we'll start this the topic of transcription. Okay, the in detail. So now this particular mRNA is formed, and then this as you know, we have exon and introns in our system, in the eukaryotic system. So what happens, this exon is being linked together and introns are removed from the RNA chain through the process of splicing. Okay, now what happens, in this particular conversion, the thymine is replaced by uracil. So the bond that is formed is uracil and adenine. The complementary base pairing is between uracil and adenine as compared to thymine and adenine in DNA. Okay, and then this primary transcript, or the first the mRNA is formed, and that is the primary transcript that can be the precursor also in eukaryotes, and that is directly used as a source of protein in prokaryotes. So now what happens, this but in this thing that we are talking about is with respect to transcription for now. And then this splicing is for the chemical, is in this chemical modification, such that the introns are removed from the RNA chain. Okay, so moving on to translation. So what happens, once this mature mRNA is formed, so various modifications are there that we'll discuss when we'll see about the RNA processing. So now what happens, mature mRNA is formed and that is being transported to the cytoplasm. Okay, now this mRNA is being used by the ribosomes to form the protein chain. Okay, so this protein chain is being linked by the uh, three base pairs that is called as codons. Okay, and here the RNA chain is being modified to form proteins in the form of proteins. Okay, and here the major role or the enzyme involved are the ribosomes that are being majorly involved in translation. So this was the major briefly the process of transcription and translation, and how the whole process works we'll discuss in the next few classes. Okay, so here transcription and translation in prokaryotes. Now we'll be discussing some things about the prokaryotes and eukaryotes. So first, let us briefly discuss about something about the prokaryotes. So let's take an example of the bacteria. This is a cylindrical bacteria that we are talking about. So what happens in prokaryotes? There is a circular supercoiled genome. Okay, and this genome is not separated in any of the compartments, and it also has some non-histone chromosomal proteins that basically helps in its supercoiling. And then we have some extra chromosomal DNA that is called as plasmid. Okay, so here we are not concerned about the plasmid part, but we are concerned about the main genetic material. And this genetic material do not have introns. Okay, these are the non-coding sequences that intervene between different exons. Okay, so this is one thing. And like as we know that the bacterial cell is a primitive cell. Okay, so what happens, to reduce the energy consumption, so all the processes that, just 1 minute. So all the processes that takes place in the system is to apply minimal energy consumption. Okay, okay, so what happens, as we can see, this transcription directly yields the mRNA. So in case of prokaryotes, as I discussed, there are new introns. So this mRNA is directly formed and without any processing involved in this case. To this is one more example to reduce the energy consumption. And then this mRNA is directly translated to the proteins. Okay, and then this transcription is being coupled with the translation. So what happens, this being the genetic material. Once the transcription starts, it forms the RNA, mRNA. And so this is the DNA, this is the mRNA. And what happens when the mRNA is formed, it is so short lived that the ribosomes attach to the 5' end of the mRNA once it is being transcribed from the DNA, and from here the proteins are also being translated simultaneously. Okay, so this leads to this degradation of mRNA after some time of its transcription. So this is called as co transcription translation, because they are coupled with each other in prokaryotes. Okay, now moving on to how it happens in eukaryotes. So as in eukaryotes, what happens, there is a particular compartment for the nucleus and the other organelles. So here the nucleus is basically the region where the genetic material is present in a very supercoiled state, and that is separated from different organelles through the cytoplasm. Okay, so this is the mitochondria, this is the endoplasmic reticulum, and then we have the Golgi apparatus, then we have centrosome, and various other organelles. Okay, so now in this case, what happens? The transcription happens, then the first thing that is formed is this is the precursor mRNA. Okay, this is not the mature mRNA because it has introns and exons that are side by side to each other. Okay, now these introns are removed and certain modifications like capping and tailing occurs on the mRNA. So this is called as processing or transportation of the processing or transportation. Then what happens? This is the then this particular this is called as mature mRNA. This flows through the cytosol outside the nucleus, and here ribosome attaches to this particular mature mRNA and forms the protein chain, that is the polypeptide. Okay, in the cytoplasm. So here there is different compartment for both RNA processing as well as the polypeptide formation. Okay, so this is what we have discussed, that is written, and this is the difference between the transcription and translation in prokaryotes and eukaryotes. So if you have any like questions related to this in your exams, you can easily distinguish between two with the help of the diagram. Okay, so moving on. So there are two kinds of genes that are present in our system. So like transcription can be a choice or can be a necessity. Okay, so many genes are transcribed all the time that are called as constitutive genes or housekeeping genes. Okay, for example, we have P53 protein or TP53 gene. Its gene is TP53, and this gene is constitutively expressed. Okay, many other genes that are involved in cell survival pathway or the cell death pathway, those genes are constitutively active, although they are regulated, and the number of copies of proteins that will be present inside the cell are always regulated. But these genes are constitutively active. The cytoskeletal genes and all those genes. Okay. Now, in for some of the genes, there is an on or off switch that are called as inducible genes. Okay, majorly the amount of proteins that we need depends upon the like function of the protein or the environment that the protein is present. So, depending on the requirement of their product. So, for example, lacoperon and tryptophanoperones, the lambda phase life cycle that we'll see later, and so these are called as inducible genes. Okay, so any external inducer or stimulus is needed to basically switch on these particular genes so that their products are being formed. And thus if the genes if the proteins are no longer needed, then these genes are switched off so that they do not produce more amount of RNA. Okay, so this is the basic difference between both the genes, that is constitutive or inducible. Okay, now we'll be starting with the major topic, that is transcription in prokaryotes. Okay, it is like in the form of proteins and other things, it's different in the case of eukaryotes. The procedure remains almost the same. Okay, so now moving on with the starting with the structure of the gene. So what happens? What we have is we have uh, like starting from the basic. First, we have the DNA. Okay, so DNA is being labeled as 5 prime to 3 prime, and all the RNA coding or DNA polymerization occurs from 5 prime to 3 prime direction only. So 5 prime means it has 5 free 5 prime phosphate group, and 3 prime means it has free 3 prime OH group. Okay, so both the strands we have 5 prime to 3 prime, and in the complementary strand we have 5 prime to 3 prime here. Now we have three things that are important for the transcription. So starting from the promoter. Okay, so promoter is basically the RNA polymerase binding site. Okay, so promoter is never the initiation of transcription, it is the binding site of the RNA polymerization. And then we have one that is called as transcription start site, from where the first base of the RNA is added. So this is called as transcription start site, that will be labeled as TSS in the further slides, and it is considered as +1 position. Okay, so here it will be +1. Before that, anything that is the promoter region will be in the minus ranges, and anything after that will be considered as plus ranges. Okay, now comes the major RNA coding region, that is called as the cistron. That is the RNA coding region from which the RNA is being coded or RNA is being transcribed. Then comes the position that is terminator. Terminator is the transcription end site. So what happens basically at the terminator position, the RNA polymerase is being snatched out or being removed from the DNA. Okay, now this is the transcription termination site where it will be separated from the DNA, and this will be the last base of that particular RNA. Okay, so these are the major three things about the transcription. Okay, and like anything before the transcription start site is the upstream, and after that it's the downstream. Okay, now as the transcription also occurs from the 5 prime to 3 prime direction. So here we, as DNA has two strands, so here we will have two strands. So this is the transcription bubble. Okay, I'm drawing like this as simplicity. So this is 5 prime to 3 prime, and this is 3 prime to 5 prime. So when transcription will occur, it will occur in only 5 prime to 3 prime direction. Okay, so what will happen, this will be the, so we will needing this as the template strand. Okay, so RNA will be started from here, this will be the 5 prime end, this will be the 3 prime end of the RNA, and this strand where the RNA polymerization starts, this is called as the template strand. Okay, and as it is always polymerized in a complementary base pairing fashion, so the sequence of this particular mRNA will be same as this strand. Okay, so this strand that has the sequence same as that of the RNA is called as the coding strand. Okay, so this is the coding strand that has same sequence as RNA. Other name of this coding strand is sense strand or non-template strand. Okay, and similarly, other name for this template strand is non-coding strand or nonsense strand. Okay, so do remember these names, although this is a bit confusing. But if you do, if you have not understood, you can ask me in the next class. And then this thing is being asked in various competitive examinations. So what they ask is, they just give the sequence of the DNA, that will be the coding strand with the 5 prime to 3 prime template, and ask you for the RNA sequence. So what you have to do is, you have to change the T in DNA to U. The sequence will be same. And for the if they give the sequence of the template strand, then you have to choose, or you have to code it in the complementary fashion and write the sequence in the 5 prime to 3 prime direction. Okay, so this is the how the questions are asked in competitive examinations, and this is the basic structure of the gene that we'll be discussing more about in the further slides. And moving on to the structure of the gene of a prokaryote, so it is arranged in the form of an operon model. This is the case for the prokaryotic gene structure, and we'll discuss more in detail in the operon uh, slides. So moving on, so as I discussed, the transcription always move from 5 prime to 3 prime direction, and this is the template strand from which the RNA is being produced, that is the complementary base pairing, and this is the sequence that is same as that of the RNA, so this is the coding strand. Okay, now what happens, one template strand of gene 1 might not be the template strand of the gene 2. Okay, so what happens, because of the directionality that is different in both the genes, as well as the nature of the RNA produced, so they might not be same in both the cases. As you can see in this particular diagram, the template strand is the below one. Okay, and here the template strand is the above one. Okay, so it might not be the same for both the genes, as it depends upon the gene to gene and the RNA that is found from them. Okay, so moving on, the basic process of transcription, how it happens. So what do we require for this particular transcription? First is the DNA template, okay, through which the RNA will be formed, and then we will be requiring the ribonucleotide 5 prime triphosphates. So we'll be requiring ATP, GTP, CTP, and UTP. Okay, then we require Mg2 plus, the RNA polymerase, and here we do not require any primers. Okay, that is required with the only in case of the DNA application. So here the primers are not required, and the RNA polymerase has the ability to start the process of transcription without any like previous nucleic acid molecule present there. And the RNA polymerase has low fidelity as compared to DNA polymerase, and it has error rate of about 1 in 10 to the power 4 to 10 to the power 5 nucleotides that are being transcribed, and it transcribes around 30 nucleotides per second, that is very much less than as compared to the DNA polymerase. So for DNA polymerase, bacterial DNA polymerase, it's around I'll check and let you know. I am not remembering the correct value, so I'll let you know about the uh, movement of the DNA polymerase. But the RNA polymerase has a less rate of base incorporation in the RNA molecule. It's 30 nucleotide per second, and it is highly regulated at each of the three steps of the transcription, that is initiation, elongation, and the termination. And the so what happens, as you can see in the diagram also, so the RNA polymerase first binds to the promoter region, and here the melting or the denaturation of the DNA starts, so that it separates out both the strands. And then what happens, this is called as the transcription bubble. Now the first nucleotide enters, and it is formed in the form of the ribonucleotide, and here the first nucleotide retains all the three phosphate groups with it. Okay, now further all the nucleotides that are joined, they only retains the alpha phosphate group. The other two groups are beta and gamma groups of the triphosphate are removed, forming the ribonucleotide chain. Okay, so this process till some of the bases are just kept, then the process of elongation. So this is the process of elongation, and then the PPI that we are talking about. So pyrophosphates are removed, and this pyrophosphates, using the pyrophosphatase, is removed to form the phosphate group, thus releasing the amount of energy. Okay, and then we have this. This is the part of the elongation followed by the termination, where the RNA chain that we have is being released from this particular DNA. Okay, and only one strand is transcribed in most of the cases, and the other strand, if it's non-coding, then it is being degraded in different steps. Okay, so this is the basic process of the transcription. And the triphosphates that we have, the nucleotide triphosphates, these basically has like these are the raw materials for the transcription, because these only will form the RNA chain. And this is the first thing, and second, they are the energy source. Okay, so this was the use of the ribonucleotide triphosphates. Then moving on to different steps of transcription. So the first one is the initiation. So this involves the recognition and binding of the RNA polymerase. So we'll in the next slide we'll talk about what is the sigma subunit, and the it is a part of the RNA polymerase. So the promoter is recognized by the sigma subunit of the RNA polymerase, and then it forms the closed complex. Then open complex is formed such that the this is the transcription bubble. So from here the transcription starts. So this is the open complex, and around 12 base pair of DNA is melted in this case. And then the first few nucleotides are NTPs are formed and joined, and this leads to the process of initiation, completion of the process of initiation. Then comes the process of elongation. This particular RNA chain is being like moved till the terminator. So the NTPs are joined, and this transcription bubble moves to this side on the gene, and it moves till the terminator is formed. The terminator is reached. Okay, and what there is a process called as abortive transcription. So what happens, like after addition of 8 to 10 nucleotides, the RNA polymerase slips down, and this particular RNAs are released, and this leads to the abortive transcription. Such this process is mainly seen in vitro, in the in vitro transcription process, but this has some implications on the fact that RNA polymerase do not transcribe when until it gets some stable interactions with the DNA. So it slips down and then releases the abortive RNA that is not the proper RNA for that particular gene. And this basically happens in vitro, that has been seen by the scientist. And the basic elongation process is seen when the sigma subunit of the RNA polymerase is released. Okay, so this release is marked as the starting of the process of elongation. And then we have the uh, RNA polymerase that translocates over the DNA template like this, as we have seen. So that it translocate over the DNA template and form the RNA chain. Okay, now and in the termination, what happens? We the transcription bubble reaches the terminator position. So here the RNA polymerase is released with the RNA molecule. Okay, so it can be in protein involved, that is the row protein that might be involved in the process of termination, or the row independent termination where the RNA molecule is being released without the help of any protein. Okay, so this was the basic procedure of the transcription that we'll talk about it in detail in the next few slides. So now moving on to the bacterial RNA polymerase. So it is a well characterized molecule. Okay, and it is a complete enzyme or holoenzyme that have four subunits. Okay, so it has two alpha subunits, one beta prime, and one sigma subunit, and there is one more subunit that is omega that helps in its assembly. Okay, so let's move on this here. So so as we have talked about, so there are two alpha subunits that have a mass of 40 kilodalton each. It has been encoded by the RPOA gene. Okay, so in bacterial system, in the prokaryotic system, we label the gene name in smalls and italicize it, and the protein name is being like used in the caps. The first letter is caps and the other ones are smalls. Okay, so this is the nomenclature how we use to write these names. And then this alpha subunit has two roles. It is involved in the enzyme assembly or promoter recognition. Okay, so it helps in enzyme assembly, and this alpha subunit is involved in the upstream promoter sequence that is being involved. Okay, recognition of this upstream promoter sequences. Then we have the beta subunit. So it is the 155 kilodalton subunit coded by the RPOB gene, and it these two beta and beta prime, these form the catalytic center of the uh, RNA polymerization. Okay, and here in these two subunits, the Mg2 plus cation is involved as a catalyst. Sorry, as a cofactor. Then we have the beta prime subunit that is 160 kilodalton, and this is the highest size subunit of this particular enzyme coded by the RPO-C gene. And along with the beta subunit, it forms the catalytic center. And then we have the sigma subunit coded by the RPO-D gene. This ranges from 32 to 90 kilodalton, and this helps in promoter specificity. And there are different kinds of different kinds of sigma subunit based on their molecular weight that recognizes different genes involved in different processes, but sigma 70 is the commonest one, and in most of it is involved in most of the bacterial processes. Okay, so this core enzyme, so we have two enzymes, one is the core enzyme and one is the holoenzyme. So core enzyme basically involves the proteins that functions as a unit. And holoenzyme is whether some subunits are basically removed from the core enzymes, leading to like when their function is being over. So the holoenzyme is the complete subunit set of the enzyme involved that is being involved in some of the processes also. Okay, so the core enzyme is the only 2 alpha subunit, beta and beta prime, and the holoenzyme is 2 alpha, beta, beta prime, and the sigma subunit. Okay, so this the total molecular weight is around 465 kilodalton. So we have two alpha subunits, one beta, beta prime, one sigma, and one more. So one more thing, that the sigma subunit has a major role in promoter recognition. So if the sigma subunit is not there, then the polymerase cannot bind to the promoter sequence and cannot initiate the process of transcription. Okay, so these are the basic roles, and the omega subunit that we are talking about, this helps in basically assembling or these core subunit proteins. Okay, so just a recap. So alpha subunit, it helps in the enzyme assembly as well as in the recognition of the upstream elements that we'll talk about in the promoter section. And then we have beta and beta prime that has a catalytic center, so it helps in polymerization, and these two are involved in binding to the cofactor Mg2 plus. Then we have the sigma subunit that helps in promoter recognition. So we'll talk more about the promoters, like what are the types of sigma subunits, and then we have the omega subunit that helps in the assembly of the RNA polymerase. Okay, so this was the idea about the RNA polymerase and different subunits involved. So this is the region of this is the sigma subunits that we were talking about. So sigma 70, and as I discussed, that these sigmas are named on the basis of the molecular weight that they are formed, that they are weight. So sigma 70 has a 70 kilodalton molecular weight, and it is involved in the recognition or transcription of most of the genes. Then we have sigma 32 that is involved in the genes involved induced by heat shock. And then we have sigma 28 that is involved in the transcription of genes that is involved in motility and chemotaxis. Then we have sigma 38 stationary phase and stress response gene. Then we have sigma 54 nitrogen metabolism and some other functions that are being under study. So these are the basic sigma subunits that have been studied till now, and based on their molecular weights. And this is the promoter regions that they recognize with the consensus sequence. Okay, so now moving on to the basic promoter region. So talking about prokaryotes, there is a consensus sequence, and so starting with the promoter sequence in the prokaryotic system, so it has two major sequences, that is the -10 and -35 region. Okay, so -10 is the place that is present at -10 position upstream of the start transcription start site. Okay, and it has a consensus sequence of TA TAAT. So it is also called as Pribnov box in prokaryotes. Okay, and then we have the -35 region of the promoter that has a consensus sequence of TTGACA. So what is this consensus sequence? So what happens, like for example, we have these bases, and in most of the organisms and most of the genes, prokaryote, I'm talking about the prokaryotic organisms. So in most of the genes, T is present at the first position. Okay, followed by T at the second position. So there might be differences in some of the genes where it's A is present or like that, but in most of the genes this T is present and followed by these particular bases. So consensus sequence is based on the most number of genes having those particular bases. Okay, so there might be differences as you have seen before also, like this particular sigma 70 genes have TTGACAT, this particular minus 35 region, but the actual sequence is TTGACA. And this but in other like in sigma 32 we have this much long sequence, and for minus 10 we have this sequence. So this particular consensus is basically based upon the most number of genes. Okay, so this is the thing. And then regarding the positioning of this particular promoter sequences, so the difference between the transcription start site and the minus 10 position is the 5 to 9 base pairs. Okay, varying in different genes. So this region, the distance is 5 to 9 base pairs. Then in the ranging from minus 10 to minus 35 we have 16 to 19 base pairs. Okay, so these two regions are being recognized by the sigma protein. Okay, so what happens in minus 35 box basically helps in closed complex formation. Okay, so it if there are mutations in the minus 35 box, then that reduce the formation of the closed complex. So it has a major role in binding of the sigma subunit of RNA polymerase. Okay, and the open complex that is formed starts from the minus 10 position. So if there are any mutations in the minus 10 position, this results in problem in formation of the open complex. So the closed complex is being regulated by the minus 35 position, and the minus 10 box helps in the formation of the open complex. Okay, and in some of the genes it has been observed that there is some upstream element called as UPE that is present. So this particular region is being recognized by the alpha uh, subunit of the RNA polymerase that increases extra specificity of the promoter. So if the promoter, we are discussing about promoters, so we have two kinds of promoters. One is strong and one is weak. So strong means they produce large number of RNA copies, and weak means they produce small number of RNA copies. Okay, so this depends upon the promoter sequences, whether the promoter will be strong or weak. For prokaryotes, most of the promoters are strong. Okay, for eukaryotes, most of the promoters are weak. So this upstream element basically increases the strongness of the some of the promoters. Okay, so this is the thing. So moving on to the process of transcription in detail, so we'll be starting with the initiation, that is the promoter recognition only. So now what happens, as we have seen, this is the minus 35 box, this is the minus 10 box, this is the transcription start site plus 1. So this RNA polymerase has bound, and this is the sigma 70 subunit that has recognized both minus 35 and plus minus 10 box. So this subunit is so big that it recognizes both of the regions of the promoter. Okay, then in the backside we have alpha subunits and the beta beta prime, and this is the omega that basically helps in arranging this particular RNA polymerase subunits. Okay, now moving on to the initiation. So this particular orange thing is the RNA polymerase with the sigma subunit, and when it binds to the promoter, okay, so what happens, it takes a region, uh, like it occupies the base pairs between 75 to 80 base pairs, that is between the region minus 55 to plus 20. Okay, so here what happens, first the RNA polymerase recognize the sigma subunit recognizes the minus 35 and minus 10 box, and this arrow represents the transcription start site. Okay, so and it binds to the promoter region and some of the region that is like after the plus one site of the transcription plus one site. And now the RNA polymerase covers about 75 to 80 base pairs on the promoter region that ranges from minus 55 position to plus 20. Okay, so this particular binding of the RNA polymerase on the promoter leads to the formation of the closed binary complex. Okay, so the closed form complex that we were talking about, so this minus 35 region helps in the formation of closed binary complex. Now, this recognition leads to creation of the tension on the minus 10 region. So what happens? This promoter region. So one thing to remember here, the promoter region that we see is 80 rich region. Why 80 rich? Because the melting of this particular region is easy and requires low energy. Why low energy is required? Because only two hydrogen bonds are involved in the complementary base pairing of A and T. If it would be GC rich, then it would be difficult for the region to be melted. Okay, so hence all the promoter regions or the regions where the melting needs to be done very frequently, hence or started to be melted, those are 80 rich regions. Okay, and so this creates a tension at the minus 10 region, and it leads to the formation of the or melting of that particular region, forming the open complex, or this is called as the transcription bubble. Okay, so here the melting starts from the minus 10 region and it leads to a particular formation of the particular transcription bubble. Okay, so that's why we said that minus 10 position helps in the formation of the open complex. Any mutation in those particular regions lead to a reduction in the open complex formation. Okay, now this is the open binary complex where the transcription bubble has already been formed. Then we have as the process increases, we have two nucleotides added on the first position and the second position. So this is called as the short RNA DNA hybrid, and only two nucleotides are incorporated, that might be A or G, so that the mm, like uh, the bonding is a particular more robust as compared to the 80 rich region. Okay, so here the first two nucleotides are formed, and this is called as the ternary complex. Okay, so this leads to the initiation of the transcription process. The first in brief that we discussed, the sigma subunit basically. hmm, So sigma subunit recognizes -35 and -10 box. It binds to the promoter, that is the closed complex. Then it creates a tension on the -10 position that leads to the formation of the open complex and the formation of the transcription bubble. Then the first and the second bases of the RNA are formed, that is the ternary complex, and these two bases are the purines. Okay, now, moving on to the elongation process. So, elongation is particularly the addition of different ribonucleotides based on the DNA sequence, and like it grows the chain till the termination site has been reached. Okay, so now what happens? The sigma subunit is released. Okay, so once 8 to 10 nucleotides have been added to that particular uh, RNA, this sigma factor is released after that, and further, this RNA chain has been like it increases in length by the addition of different ribonucleotides and reaches the terminator site. Okay, this polymerization is being done by the core enzyme only and not the holoenzyme. So basically, here the core enzyme involves alpha to beta, beta gamma subunits. So these subunits are involved in the polymerization of the RNA chain. Okay, and here the E. coli elongation at 37 degrees Celsius is about 40 nucleotides per second. And here the concept of abortive transcription comes. So what happens once the like sorry so what happens once the transcription bubble is formed, the first few nucleotides are added. So what happens in if the sigma subunit is released after that, the polymerase is slipped, releasing these 8 to 10 nucleotide long RNA subunits. Oh, sorry, RNA chain. So, to keep this, the sigma subunit basically uh, um leads the RNA polymerase till few more bases and then it is released further so that if the RNA polymerase can be moved to the proper formation of the whole RNA as compared to the abortive transcripts. Okay, and as we know, if the helical region is being opened in this way, there will be some tension that is being formed at the uh, ends. So what happens, this is the like this. So there will be tension created here at the end as well as in the this end also. So what happens, this tension is being released by the uh, this ahead of the RNA polymerase. Here is the RNA polymerase, and this tension ahead of the RNA polymerase is released by RNA gyrase, and this like the tension that is behind the RNA polymerase is released by the topoisomerase 2. Okay, so these both the proteins functions in cut and then join manner. So they cut to release the tension and then they join again with that particular strand. So this was the process of elongation of the transcription, and then we comes at the termination step of the transcription. So it is basically dependent upon a protein called as rho protein. So if a rho protein is involved, it is called as rho dependent. If rho protein is not involved, it is called as rho independent. Okay. So in case of row dependent transcription, a row protein is involved. So that is basically a ATP dependent RNA stimulated helical protein. Okay. So and it is a homohexamer. So this, as you can see in this particular figure, the row proteins has six uh, subunits that is of the same size, and we have a rope binding site on the RNA itself. So using ATP, it binds to this particular row binding site on the RNA, and like it binds here and then moves along the RNA chain where the polymerase is moving, and using ATP it helps in releasing this particular RNA molecule by winding it up upon itself so that it can basically snatch it from the DNA, and after that the polymerase is released. Okay, so this is how the row dependent termination works. This is more clearly understood in this particular diagram. So we have a row binding site here. Okay, so here we have a row binding site. This is the elongation process that happens, and here the row protein that you can see, it's a homohexamer. It recognizes this particular site on the RNA itself. Sorry. So now what happens? It moves to the, like after recognizing, it moves to the termination site or the RNA polymerase where it is transcribing that particular RNA, and then it pauses at the terminator position. So when a row protein basically moves on the RNA chain, its um, speed is more than the polymerase. Okay, so that it can catch up the polymerase on time before it reaches the terminator position. And then what happens, this row protein basically like snatches the RNA from the polymerase and helps in the release of the polymerase, and it also helps in breaking the RNA DNA hybrid. So the complementary base pairing is being removed by the hexamer, that is the row protein, so that the RNA polymerase can easily leave the RNA molecule. Okay, then what happens after reaching the termination site? The RNA polymerase is also released from the DNA, and with the release of the row protein also. Okay, so this was the basic idea about the rope dependent termination. So here ATP is involved majorly because the movement of row protein on the RNA requires ATP. So here the ATP is involved. Now we'll see about the row independent termination. So moving on to the row independent termination. So here no protein is involved, and this is called as a intrinsic termination because it involves only the formation of a hairpin loop. So intrinsic, the RNA itself terminates upon itself. Okay, and a hairpin is formed in the RNA, and it consists of a stem loop followed by a U-rich region. So here is the structure of the hairpin that we are talking about. So what happens, this is a GC rich region that has like complementary base pairing upon itself, and like this is a stem loop kind of structure that is being formed and followed by a U-rich sequence. So this is the 5' end, this is the 3' end, that is the like uh, the region near the terminator sequence. Okay, so now what happens, this is this stem loop helps in basically uh, slowing down the polymerase activity, and then this U-rich region is the region where the sequences or the uh, like we discussed about the opening of the promoter sequence. So like this, the complementary base pairing between A and U are is really less. So this helps in breakage of this particular hydrogen bonds and the RNA molecule is released. Okay, so what happens when this terminator sequence is formed? Here the hairpin loop is formed. Okay, this leads to the stoppage of the RNA polymerase upon it upon the RNA chain, so that the RNA polymerase cannot move further. And now what happens, then the urate sequence comes that have a base pairing, less base pairing between themselves. So this helps in breaking this particular bond and the release of the RNA molecule. Okay, so although this particular procedure, that how RNA is being released with the row independent row independent termination, this is under study that how this happens, but this is the proposed mechanism that might occur in this particular case. Okay, so this is how the row independent termination works. And this now when this particular transcription bubble cannot move further, so what happens, the RNA molecule is released, the RNA polymerase is also released, further relieving the transcription bubble. And this transcription bubble is also relieved by the help of the topoisomerases. Okay, so this leads to the end of the process of transcription in prokaryotes. Now, some further extra information about the transcription is that they, we develop certain antibiotics that are related to the transcription in prokaryotes, so that they can be used in treatment of various diseases. So, first is rifampicin. Okay, so rifampicin is basically a molecule that inhibits the polymerase subunit of the RNAP. Okay, so it inhibits beta and beta prime subunit. So it basically inhibits elongation. So what will happen? As you know, elongation is being done by the core enzyme. Okay, so where this beta and beta prime subunits are involved. So when this particular antibiotic binds to these subunits, then the RNA polymerase cannot move further on the transcription bubble. Okay, it will be stopped. This is how this particular antibody functions. And then we have actinomycin D. So it's basically a phenoxazone ring-like structure. It has a phenoxazone ring-like structure that gets integrated into the GC-rich region of the DNA helix, and it results in complete distortion of the helical structure, such that both DNA and RNA polymerase cannot bind. So, as you have seen, in the -35 region, there is a GC-rich region. So, what will happen if this particular antibiotic binds to this particular -35 region? So, what will happen? The distortion in the helix domain of the DNA will not be helpful for the RNA polymerase to bind. So as we have discussed previously also, when the -35 region might have been mutated, then that would reduce the formation of the closed complex. So sigma subunit cannot recognize this particular uh, -35 box, and hence RNA polymerase cannot bind to the -35 box, hence the closed complex cannot be formed. Okay, so this is how actinomycin D. So if we can see, so this particularly for inhibits initiation. And not only RNA transcription, but this particular antibiotic also targets the replication process also. Okay, so using the same phenomena, because DNA polymerase also helps, also is like bind to the particular region, that is the origin of replication in the genetic material. And it also requires certain recognition sequences that GC rich, and in this case, if actinomycin D binds to that particular region, so our DNA polymerase also cannot bind to that particular recognition sequence. And the third antibiotic is cordycepin. So this is the structural analog of adenosine. So adenosine means we have adenine group as a base. So this is the structure. We have adenine, and this is the OH. So this is 2 prime, 3 prime, 4 prime, and 5 prime OH. This is the phosphate group involved. So here the 3 prime OH is not present. So this OH is basically important in nucleophilic attack. For polymerization, this particular OH is very important. So if this particular OH is not there, hence the polymerization cannot happen. So what happens when cordycepin is involved in replication, so it binds to the in place of adenine, and it stops the elongation or the polymerization of that particular RNA chain. Okay, so this also targets elongation process like rifampicin. Okay, so these were the three antibiotics. The role of these antibiotics have been asked in various competitive examinations. That why I included here so that it is clear the mechanism, how it functions in this particular case. And the further we'll discuss some of the experiments. How can we design experiments in binding of proteins to different RNA molecules or like DNA molecules. So thank you, and if you have any questions you can ask me in the next class.